Hello, my name is Cash Merrill, and this is my exam to write on. So let's go ahead and get started. Number one is talking about the autosomal dominant polycystic kidney disease. So it tells us that the disease allele is dominant, so I made it P in our Hardy-Weinberg equation, and the non-disease allele is therefore recessive, so I made it Q. So it's asking, what is the frequency of our non-disease allele? So if 1 in 750 people are infected with the disease, which is dominant, then we know that to get our Q squared, we put 749 people over 750, gives us our Q squared, which gives us 0.99867. Now to get Q, we take the square root of that number, and that gives us 0.9993 for our Q or non-disease allele frequency. Now part B says, what is the frequency of the ADPKD, or the disease allele in the human population? So now we just use the P plus Q equals 1 equation. So P plus our Q, which is 0.9993, equals 1. Subtract that, we get 0.0007 for the frequency of our ADPKD allele. Now letter C asks, what is the frequency of heterozygous individuals in this population? Well, the frequency of heterozygous individuals is 2 times P times Q. So 2 times 0.9993 times 0.0007 gives us 0.0014 is the frequency of heterozygous individuals in this population. And D is asking, out of a population of 1 million people, how many heterozygotes are expected? So we just take that number, our frequency of heterozygous individuals, multiply it by the population size of 1 million, and we get 1,400 expected heterozygotes in this population. So number one, pretty simple. I'm just going to move this over a little bit for number two here. So number two is talking about a DNA molecule. So it gives us in the question that it is a DNA molecule. And so part A is asking, there's all these labels, so W, X, Y, and Z on the diagram, and it's asking what these indicate. So W on our picture here, I've attempted to draw it, I'm not an artist, but W here represents our phosphate group as part of the DNA backbone. The letter X here, pointing to this pentane ring, is our pentose sugar, and in this case, since it's DNA, it's a deoxyribose, a 2-deoxyribose pentose sugar. And then our letter Y, pointing here at this nucleotide, since it's paired with A, which is adenine, it is thymine, because A pairs with T in DNA. Letter Z down here, so up here in the picture, these are kind of the same color here. So C pairs with G always, so cytosine pairs with guanine. So we know that C is a guanine nucleotide, or a guanine nitrogenous base, excuse me, these are nitrogenous bases. So Z is a guanine. B asks us to indicate the 5' end and 3' end of each DNA strand. So the 5' end is where the phosphate group is. So this is the 5' end of this backbone, and the 5' end is on the opposite side since they run anti-parallel. 5' end is here, and the 3' end is where the hydroxyl group will stick out off of this third carbon. And it's the opposite on this side since they're anti-parallel. Letter C asks us to indicate the portion of the molecule that is considered the backbone. So I've attempted to circle this right here. So it is the sugars with the phosphate group. So the deoxyribose sugar with the phosphate group makes it the backbone. And so it's like the ladder, the size of the ladder, and then the nitrogenous bases make up the rungs of the ladder. And letter D on the image above indicate the portion of the molecule that makes the DNA, gives it a net negative charge. So these phosphate groups give it the negative charge. All these phosphate groups give the DNA molecule its negative charge. So now let's move on to number 3. Number 3 is a longer problem. So number 3 is our problem about the 3 linked genes in the fruit fly Drosophila. So we have kidney bean shaped eyes, which the K is the recessive allele and the K plus is the wild type, and that's how it is for all three, for the ebony body color and also for the cardinal eye color. So it's saying that we're crossing a female who's heterozygous for both traits, so that's right here, K plus E plus CD plus and KECD, so she's heterozygous with all three, to a homozygous recessive male, so he has no wild types. So he is, so no wild types. He is kidney-shaped, ebony, and cardinal for both chromosomes there, because it's homozygous recessive. Now it's asking us to determine the allele arrangement. So we have our cross here, and then on our list of the observed outcome of the cross, I found our parental crosses, which were the two most, and so they're two most like the parents. So the parental cross was a kidney-shaped cardinal wild type, so that was 1773. Then we had a wild type ebony body, and that was 1761. So that determines our allele arrangement. So we know that we have the one wild type on top with cardinal and kidney-shaped eyes, with wild type ebony body on bottom. Now we want to determine the order, what order these would go in. So now I drew up the three possibilities that we have, with CD with the cardinal in the middle, the kidney shaped in the middle, and with the wild type E in the middle. Now, then I found the double crossover offspring on the list, and so one of them was a kidney-shaped wild type ebony, and the other one was a wild type cardinal wild type. So I went through and looked for which of these combinations would give this on a double crossover. So for example, we have kidney, wild type, ebony, wild type. So this one doesn't work. Here we have cardinal, so here we have kidney, wild type, and ebony. So this one works. Here we have kidney, ebony, cardinal. So this one does not work. So we determined that this one is correct. So that is our gene order. We have cardinal, kidney, wild type, with wild type, ebony on bottom. So this is our correct gene order. Now we want to discover how far away each of these genes are on the chromosome. So we want to make a linkage map. So in order to do that, I wrote up a new cross here with the correct order, crossing it with another homozygous recessive male. Now we get, first we'll determine the distance between the cardinal and the CD and the K alleles. And to do that, we use our double crossovers again here. And then we need to just switch just the first, so just our cardinal with our wild type, so just the CDs, we're going to switch them, which gives us a wild type, kidney, wild type, and a cardinal, wild type, ebony. So when I looked on the list, those totals for those offspring were 97 and 89. So I add those up, divided by the total number of offspring, which is 4,000 times 100, gives me 5 map units between the CD and the K alleles, between the eye shape and the cardinal's eye color alleles. Now we have to find the distance between the eye shape and the body color alleles. So we do the same thing. We have our double crossovers, and then when we just switch our ebony here with wild type and the ebony, we get 138 and 128 offspring who have those phenotypes. So we add those all up, 8 plus 6 plus 138 plus 128 divided by 4,000, the total number of offspring, times 100 gives us 7 map units between the body color and eye shape alleles. So now we can make a linkage map. So we have our cardinal, so our CD plus CD is 5 map units away from our one in the middle, which is our K plus and our K, which is eye shape, which is 7 map units away from our body color E plus E. And that gives us our linkage map right there. That is the order and also the distance between each trait on the chromosome, each allele. Now, between each gene, excuse me. Now we want to calculate an interference value. So we take our map units, each divided by 100, multiplied together, gives us 0.0035. We multiply offspring to get our expected number of double crossover offspring, which is 14. Now we add our observed number of double crossover offspring, 8 plus 6. It also gives us 14. So when we plug it into our equation, I equals 1 minus the observed number of double crossovers divided by the expected, it gives us 0%. So we had nothing interfering with what we expected and what we actually observed for the number of double crossover offspring. And so that's it for number 3. Moving on to the last problem. Number 4 is talking about another disease, another syndrome, Higashi, or Shidiak-Higashi syndrome. I'm not sure how to say it, but in this time it's an autosomal recessive disease. So it's similar to problem 1, but now we're dealing with autosomal recessive disease. So this time our disease allele is our recessive gene, which is Q, and our non-disease allele is our capital, which is P. So if we want to find Q, it says that it affects 1 in 32,000 people. So Q squared equals 1 divided by 32,000, which is 0.00003125. Now to find Q, we take the square root of that number, which gives us 0.0056. That's the frequency of our lowercase d, or our disease allele in the population. Now to find the frequency of the capital D allele, which is the non-disease allele, we go P plus Q equals 1, so P plus 0.0056 equals 1. Subtract that from 1, we get 0.9944 for the frequency of our non-disease allele, or the capital D allele. Now it's asking us, what is the frequency of a homozygous dominant genotype in this population? So a homozygous dominant genotype is P squared. So we know our P is 0.9944. So if we just, this is right here. 0.9944 squared. If we do that, then we get 0.9888 for our frequency of a homozygous dominant genotype in this population. Now letter D is, what is the frequency of a heterozygous individual, heterozygous genotype in this population? So that's capital D lowercase d, which is 2pq. So 2 times pq is 2 times RQ, which is 0.0056, times p, which is 0.9944, and that gives us 0.0111 as our frequency for a heterozygous genotype in this population. So that'll be it for the questions for this exam 2 write-on, and thank you for watching.